Hi guys, I'm Ashish and this is my channel MYCOBACTERIUM India, and I welcome you back to my channel. And today is the second day of the series of top 25 countdown for life sciences. Here I am going to discuss 25 questions from the biotechnology discipline. So guys, this video will be beneficial for all the students, all the aspirants who are preparing for like CSIR, ICMR and DBT-JRF, moreover MSc and PhD entrances, and last but not the least, the NEET students. So guys, let's continue with the questions. So guys, first question is, which one of the following is a non-parametric test? So guys, this question is from the biostat. So guys, as I said, ki biotechnology is a very vast discipline, and biostat, bioinformatics, all these are very important in the entrances which are related to biotechnology. So guys, first of all, you can see the option is A. Option A is T test, B is Z test, C is F test or ANOVA, and D is the chi-square test. So guys, there are two types of tests. If you are have knowledge about the biostat, then you know we have two kind of tests. First is parametric and second is non-parametric. Parametric is where there is the normal distribution of curve is followed. But what happens in case of asymmetrical, like you can say probability distribution, there exists the non-parametric test. And when we talk about the non-parametric test, chi-square test and U test are very important. So guys, the answer is D, that is chi-square test. So when the question like this comes, which of the following is a non-parametric test, you are just going to take chi-square with a blind eye. So guys, the second question is, which of the following is the most abundant amino acid? So guys, here it becomes very important to remember which is the rarest and which is the most abundant amino acid. So guys, if we talk about the rarest, that will be the tryptophan, and if we are going to talk about the abundant, most abundant, then it is the leucine. So guys, option number D is correct. So guys, let's talk about RNA interference. So my question number 3 is, RNA interference was discovered by which of the following scientists? So guys, Sekman and Neher, Fire de Mello, Peter de Mello, and Robert and Calvin. So guys, this type of discoveries are very important, where some technique like are discovered by the scientists are asked. So A is Sekman and Neher, who discovered patch clamp, Fire and de Mello, who discovered RNA interference actually. So the answer is B, or B is the correct answer. And C is the Peter de Mello, this is nothing relevant, and D is nothing relevant. So guys, the answer number B is correct for question number 3. For question number 4, which one of the following is an example of secondary database? So it becomes very important when we are going to talk about which is the secondary database and primary database. This type of question may be asked in the exam. And suppose, first of all, I would like to explain what is database. Database is the system, is the server where you can find the data about the genome or the protein of the individual. What happens in center dogma, there are three things which are important. That is, first is the DNA, RNA and protein. So guys, what happens in bioinformatics? Bioinformatics there are several databases which are important. What happens? One is the molecular basis of inference, and second is what? Second is the dry lab, where you can find the bioinformatics. What happens when sequencing was discovered? Then it came into existence that we have to make a database, like database of all the genome, all the gene, all the proteins which are encoded by different codons. So it becomes very important. So guys, what happens when such type of questions are asked, where secondary, primary or composite database example are asked, you have to remember all the things. First you have to understand what the primary database is and what secondary database is, and what is the composite database. Primary database is that which has actually the actual database, actual discoveries by the scientists are kept. And what happens when those are copied in the second database, that database is known as secondary database. Correct answer is PROCIT. PROCIT is nothing related with the primary database. That is secondary database. GenBank, SwissProt and PIR, Protein Information Resource. These all three are what? Primary database. So guys, question number 5 deals with, so guys, as I said to all of you that not the chemistry part is important, but it's very important. So what happens here, you will see the numerical quantitative assessment type questions where like molarity and normality questions are asked. So guys, very important to prepare all those chemical analytical questions. So guys, 1 m sugar solution represents 1 gram. This is very basic question, and these type of questions you have done in like class 11th. So guys, just go ahead, and A is 1 gram sugar solution in 1 liter of solvent, 1 gram sugar solution in 1 kg of solvent, 1 mole sugar in 1 liter of solution, and 1 mole sugar in 1 kg solvent. So guys, it becomes very important where you have to take the right answer, and these are very crucial and very scoring question, you can say. As you all know, aims and all the entrances are 1/3 negative marking. So guys, please do carefully all these type of questions which can be confused with like these terms, solution and solvent, and like mole and what gram. So please do carefully. Here the answer is C, which is 1 mole of sugar in 1 liter of solution, because molarity is m, and molarity always tells about the solution of the, like you can say, volume of the solution, and volume is what, in liter. So it can be said that the volume can be what, changed, but that is kg, that is weight, cannot be changed. So guys, volume can be changed, molarity can be changed. Correct answer is C. So question number 6 deals with which of the following is targeted by scientists for making corona vaccine. So guys, coronavirus has emerged as a pandemic in COVID-19 and corona questions can be asked in the MSc entrances, PhD entrances, CSIR, ICMR, DBT and NEET. So guys, very important to know which is the corona vaccine target element. So if you will go to the details of the coronavirus structure, then what happens here, section like A, B, C and D are 4 options. B, C and D are very like you can say unnecessary thing. A is spike protein, which is the very correct answer. So guys, A is the answer for question number 6. So guys, let's come to the 7 number question, which tells about why gene of lac operon is responsible for what. So guys, as I discussed, if you go back to my lac operon topic, you will see that I have given a trick to remember, that is ZYA, Z-Y-A. These 3 are the genes present in the lac operon, ZYA. Z for what? Galactosidase, beta galactosidase. Y for permease, and A for transacetylase. Transaminase is nothing with this like gene, this operon system. So guys, correct answer is B, that is permease. Question number 8 is which blood group people are universal acceptor. These type of questions can be asked, but rarely asked, but can be asked. So prepare all the things with the immunology, like these type of questions come in immunology. So guys, first is O positive, O negative, AB positive and AB negative. So guys, here you have to remember 2 things. First is universal acceptor and second is universal donor. Universal donor is O minus, as you all know, and AB plus counterpart is the universal acceptor. So guys, question number 9, biotin is which vitamin? What is the nomenclature of vitamin like biotin? So guys, B10, B9, B7, B3. So this also is very typical question, can be asked several times and can have asked many times in the examinations. So guys, B7 is biotin, B3 is niacin, and you must have to remember all these like two things that is frequently asked in the examination. B7 and B3. B7 is biotin, B3 is niacin. So answer is C, and C is the correct answer. Insulin signaling is done by, I mean when insulin binds, which is the signal, which is the pathway that comes into existence. So guys, answer number A is JAK state pathway, B is G protein coupled receptor, C is MAP kinase pathway, and D is none of these. So guys, this question from cell signaling or transduction. So you have to prepare cell signaling and cytology very carefully, because these types of questions are very frequently asked. So guys, C is MAP kinase pathway is the answer. This can be possible if you will study. If you will not study, then you are not going to able to handle this type of question. So please study not in deep, at least from periphery of these topics. At least touch the topic and know what is the status of the question. What I mean which question can be important, like epinephrine ke signaling pathway they can ask you, and like IP3 DAG pathway, all these things, calmodulin pathway, these are very important pathway, and you have to know all the rules of primary and secondary messenger like camp. So guys, please be prepared with all these things, primary, secondary messenger, and third thing is transmembrane receptor for each ligand. These should be clear, and the nature of the the binding ligand. So guys, question number 11 is tonoplast is related to which of the following organelle? A is mitochondria, B is Golgi apparatus, C is vacuole, and D is lysosome. So guys, tonoplast is a special kind of membrane, and it is in like vacuole. Vacuole is bounded by what? The tonoplast. Tonoplast has a capacity like to like shrink and contract. It is a very specific property of the vacuole, and so the C is the correct answer. So question number 12 tells you about genome of which of the following bacteria was sequenced first. This is very, very, very important. How many very I can say, I will say to this question. So guys, the A is Haemophilus influenzae, Mycobacterium tuberculosis, Salmonella typhi, and E. coli. So just guess and do the correct answer. Everyone is like confused with the D, that is E. coli. But this is a misconception. The first genome when the sequencing came into existence was done for Haemophilus influenzae. That is the answer is A. So A is the correct answer, Haemophilus influenzae. So guys, question number 13 is what is the carrying capacity of the COSMID? So every carrying capacity, like carrying capacity say I mean ki how much amount of DNA they can ligate, like they can insert in what? In the as a vector. So guys, A is 40 to 50 kilobase, B is 100 to 120 kilobase, C is 5 to 10 kilobase, D is 400 to 500 kilobase. Please guys, please, if you are going to appear in biotech exam of any of the exam of where the biotech is important, please at least read all the vectors, their carrying capacity, and their like gene of special genes present in those vectors. So guys, the answer is A. It is very direct question. The answer is A. COSMID can carry like 40 to 45 kilobase pair of like what? Insert. So 14 number question tells you about which of the following restriction enzyme gives the blunt end. Blunt end sticky ends are very important. When suppose a restriction enzyme like ACT, what they do, they either give a blunt end or a sticky end. Sticky end can be called staggered end. So guys, sticky end has an overhang, blunt end doesn't have an overhang. So guys, answer options are A, hind 2, B, hind 3, C, equal 1, D, BOM H1. When you are not going to know, you will be confused, because this is very, very important, very, very confusing question too. Very, very important question this is. For the CSIR point of view, NEET point of view, and for MSc and PhD entrances. Even in the assistant professor exam, they ask such type of questions for zoology or biotech if you are appearing. So guys, A is the correct answer, hind 2. This is direct question and nothing to explain. Which of the following is not a stop codon? Very, very important guys. Several times they have asked in what? The NEET exam and NET exam also. So guys, in MSc entrances also moreover. So guys, very easy trick. If you know my name, what is my name? My name is Ashish Gupta. So guys, one thing, what you do, you just take A from my Ashish and G from my Gupta. And what you do, you just take U common from all the options. If such type of options are there, take your U common. Then what you do, A has, and now see the second and third codon of the base of the codon. So here the first has GG, except if you are like ignoring U, then GG. GG means Gupta. Doesn't mean. If you this is a simple trick I am telling about. So B is AA. AA is in my what name? Ashish. है ना? And the C is UGA, Again you ignore U, and GA means Gupta Ashish, and D has UAG, Ashish Gupta. So guys, everything makes a sense for my name except for what? UGG, where Gupta Gupta. I can't be Gupta Gupta. What? But AA I can be, because my name starts with double A. So guys, here you ignore UGG. This is a simple trick. If you are going to remember, you will be benefited, because I have from my like NEET to till now PhD, I have remembered this trick. So guys, it's very important. So guys, the correct answer is A, that is UGG. So question number 16, which of the following is a filamentous virus? Or you can say filamentous bacteriophage. So guys, answer is A, T4, B, M13, C, lambda, and D, TMB. So guys, this is very important, and very important because these act as what? The carrier, that is the vector. So it becomes very important to know the carrying capacity and also the genome in the virus and the nature of the virus. So guys, the correct answer is B, that is M13. M13 are filamentous virus. Please remember these types of questions. These are very important. So guys, there is nothing explanatory is this question in this question. So everything is like in the direct form in this question. So question number 17 is, how many ATP are generated by in vivo lactic acid fermentation of one molecule of glucose? So this is again the direct question, and this question is from what? The biochemistry. Biochemistry is very important for every exam, from NEET, where this is the biochemistry known as biomolecule and plant physiology, and to NEET, where it has a separate like discipline, that is plant physiology and biochemistry. So guys, answer is A 2, B 16, C 32, D 38. So guys, very irrelevant all the options except for A. So the A is the correct answer. 2 molecules. So how much volume of water is required to make 100 ml of 1x saline from 10x? So guys, these types of questions are also asked in the aptitude in the JRF exam, and also asked in MSc entrance as analytical question, and in NEET as biochemistry question, or can be in the chemistry part. So guys, A is 100 ml, B is 90 ml, C is 10 ml, and D is 1 ml. So guys, it is very important to remember this formula. Like these formulas are very important, which are in the like chapter 1 of the 11th class. Just easy and go through that chapter 1 of the 11th class, and remember the formula for normality and volume matter, and that molarity. So guys, if you remember here what happens, here apply the M1 V1 equal to M2 V2. So guys, molarity for 1, that is 1x, can be multiplied with the 100, that is the corresponding, because what they want 1x of 100 ml. They want 1x of 100 ml. So guys, 1 into 100 equal to what? 10 into x, because you are going to take out the volume of 10x. Suppose x volume of 10x. You just write down, you will find it. So guys, 100 into 1 equal to 10 into x. 10x equal to 100 simply. So x equal to what? 10 ml. But that x is what? That x is the volume of 10x saline. So what? To make 100 ml, you have to what? Minus that 10 ml from the 100 ml. So 90 ml of water plus 10 ml of 10x will give you what? The 1x saline. So guys, 90 ml is the correct answer. Just you solve, you will get it. Just follow the instruction I have given you. Just remember the formula, all the formulas, M1 V1 equal to M2, and molarity, normality, all the equivalent weight for all the like ions, you will find it. So very easy. So question number 19, which of the following interleukin is related to parasitic invasion? Very important, very direct question from immunology, and the answer is D. IL-5 is the only thing which has like relevance in parasitic invasion. Eosinophils also you have to remember here. Eosinophils are very important in parasitic invasion. So guys, temperature of liquid nitrogen is, this question is like, you can say ki takiya kalam of any of the exam. So minus 196 Kelvin, minus 190 degree centigrade, 1 minus 96 Fahrenheit, and minus 96 Kelvin. So guys, you have to remember what is the temperature of liquid nitrogen, and answer is what? The answer is B, minus 196 degree centigrade. Nothing to explain here. SCID is, you have to just what abbreviation is given and you have to find the full form. Several combined immunodeficiency, severe compromised immunodeficiency, and so funny, severe combined immunodeficiency, several compromised immunodeficiency. Just see what is the play, what is played with the like words. So the answer is C, severe combined immunodeficiency. Nothing to remember, just remember severe combined immunodeficiency. These types of abbreviation you have to learn, you have to get by heart, and nothing to like, you are not going to use your brain here, just use your like key you have like remembered, and just flow, just in flow you are going to answer. Which of the following TLR is situated on the endomosomally, that is endosomal compartment. So guys, TLR are very important in, has like role in what? Immunological response. So guys, immunological response में TLRs have role, and TLRs are present on the plasma membrane, that is outside part of the compartment like cell, that is the you can say the cell membrane, and other they are present either on the like endosomal like membrane. So guys, TLR3, TLR2, TLR1 and TLR4. So what happens here? TLR3 is the answer, that is TLR3 is found on the like endosomal compartment, otherwise 1, 2, 4, 5 everything is found what in the cell membrane. So guys, 3, 7, 8, 9, just see what are the TLRs which are present. You can Google it and you will find they will be like 3, 7, 8. 3 TLRs are present on the endosomal compartment, otherwise all are present on the plasma membrane. So guys, just memorize this. TLR3 here is the correct answer, that is A. Dolly is a transgenic: buffalo, cow, pig, sheep. Very right question, very funny question, I must say. The D. Many times people are confused that buffalo, but not buffalo, not the pig, but what it is, a sheep. Lycopene is abundant. These types of questions are very important in biotechnology exams, even in the NEET and like NET also. So guys, tomato, potato, honey, red ant. Nothing to use your mind, just take A, that is tomato. Tomato has lycopene, so that is red. What is the pH of lysosome? The last one, question number 25. What is the pH of lysosome? 1,573. Just answer. So guys, the correct answer is B, that is the 5. 5 to 5.5 is the lysosomal pH. So guys, this is the end of the session, and I just want to thank you for the love you have given to my channel. And I must say, I am not forcing you, but this time I am telling, please subscribe my channel, share my channel to the needy ones, and please like my video. So guys, hope for the best and all the best to all the aspirants, and hope you enjoyed my video. Just like and share to your friends. Thank you so much guys.